Good morning, everyone, or good afternoon, depending on where you're coming from. My name is Karen Simpson, and I'm the Senior Manager Laboratory Services at the Australian Water Quality Centre. I'm very excited to be hosting our webinar today, entitled on solving water quality issues using the latest testing techniques and technologies. A big welcome to everyone who is attending our webinar from locations across Australia and around the world. First of all, I'd like to begin by acknowledging the traditional owners of the lands on which we meet today. And I would also like to pay my respects to elders past and present. As we know, water quality scientists play a very critical role in the supply of safe, reliable water and wastewater services to the individuals and the communities who rely upon them. They achieve this by providing a broad range of analytical, research and consultancy services associated with the chemistry, microbiology, biology and ecology of waters, tailored to all aspects of the water cycle from catchment to tap to wastewater, reuse and alternative sources. In our webinar today, we're going to be exploring the latest techniques and technologies available to water utilities that enable them to firstly monitor the effectiveness of the protective barriers designed to prevent the entry and transmission of contaminants into our drinking water systems or their discharge into the environment. Also, that help us to respond to and resolve water quality issues, and importantly, arrive at fully informed risk management processes. We're very excited to have assembled a really fantastic panel of experts today, and some have some really engaging content to share with us today. So really extend to you all to take advantage of the opportunity you have today to tap into the expertise that we have. Now, before we get going, there's a few housekeeping issues. And while your lines are all muted and your views are turned off, you can lodge questions in the Q&A box on your Zoom window. So if you go to the bottom of your Zoom window, you'll see a Q&A box there. And we encourage you, as we're going along and the presenters are presenting, just to pop your questions in there. And at the end of all of the presentations, we'll be having an interactive Q&A session where we'll come to the questions. Now, if at any stage through the webinar you experience any technical difficulties, please use the chat box and message AWA support, and they'll try to address the issues. We also advise that the webinar is being recorded today, and that's so that we have this for future reference. Okay, so that's the housekeeping items out of the way. So without further delay, I'd like to introduce our presenters. So first up, we have Dr. Torsten Mosic, who is the Manager of Field and Life Science Services at the Australian Water Quality Centre. We also have Karen Rouse, the Chief Executive Officer of Water Research Australia. Dr. Brad Clark, Senior Lecturer, Environmental Science and Analytical Chemistry at the University of Melbourne. Dr. Barbara Drigo, who's a Senior Research Fellow and Assistant Professor in Microbial Ecology at the University of SA. And we also have with us Professor Erica Donner, Research Leader at the Future Industries Institute at the University of South Australia. So I'm now going to hand over to each of the panellists just to briefly introduce themselves and provide a little bit of detail about their subject matter. So Torsten, if we can start with you.
Yes, thank you, Karen, and hello, everyone. My name is Torsten Mosic, and I have been with SA Water for 14 years, and 7 of those at the Australian Water Quality Centre. I'm responsible for the operation of the Field and Life Science Services team, which includes AWQC's Field Services Group, the Microbiology, Biology, Biomonitoring, Product Testing, and Molecular Services Laboratories. And my presentation today will be touching on solving water quality issues using our molecular analytical technology here at the AWQC.
Thanks, Torsten. Karen, over to you.
Hello, everybody. It's a pleasure to be here today and speaking. I'm not sure that I quite sit in the expert category, but I'm CEO of Water Research Australia, which is a member-based organisation dedicated to supporting healthy people, healthy lives and a healthy planet through collaborative research. So some of you will know that Water Research Australia evolved from the CRC for water quality and treatment. And so we have a 20+ year history of supporting the water industry with timely knowledge to solve their problems and take opportunities. Today, I'm going to be speaking about the importance and the value that can be derived from engagement with end users as we go through these processes of developing new techniques and new technologies with the capability, the wonderful capability that's emerging. So that's what I'll be touching on today. Thank you.
Thank you, Karen. Brad, over to you.
Hi, and thanks very much for the to be part of this panel. It's great to be here. So I'm from the University of Melbourne, and I'm the chief investigator for the Australian Laboratory for Emerging Contaminants. We're an industry aligned and partnership with, and we collaborate with water utilities and contaminated land consultants, and also our major partners are Agilent and Eurofins. We use cutting edge technology to answer some the tough questions related to environmental chemical pollution. In particular, we look at the environmental fate, impact, and monitoring of emerging contaminants.
Thanks, Brad. And Barbara.
Hi, I'm Barbara Rodrigo, and I am a lecturer at the University of South Australia at the Future Industry Institute. And in the Future Industry Institute, what we do, we use molecular tools to manage emerging risk in wastewater and water products. And this will be also the title of my talk today. And we collaborate extensively with SA Water, Water Research, and several water utility around Australia.
Thank you. Thanks, Barbara. And as I mentioned, we've got Erica with us as well from the University of South Australia. Barbara will be presenting, but Erica will be online for the Q&A session. So Erica, if you'd like to introduce yourself.
Yeah, thank you. Yeah, as you mentioned, I'm just here for my general enthusiasm of this topic. I work closely with Barbara, who runs our lab program for using molecular tools in Future Industries Institute. And my research focuses on chemical and microbiological risks in the water cycle. And I'm particularly interested in understanding the risks, the opportunities, and the best management practices for managing antimicrobial resistance in waters, foods, and agribusiness, basically. Thank you.
Thank you, everyone. So as you can see, we've got an amazing group of panelists here today. So without further ado, we're going to kick off now with Torsten's presentation. So Torsten's going to present to us some information about how DNA testing techniques offer water utilities an opportunity to improve their water quality management.
Thank you, Karen. The Australian Water Quality Centre is a leader in the field of microbiological testing, and we have a great track record of developing and validating effective testing methods for the detection of waterborne pathogens. And one of these is our molecular analytical technology. And today I'll be providing you an overview of AWQC's molecular analytical services, including the use of advanced next generation sequencing, or NGS, DNA testing methods, and they're used to optimize water quality management. Our molecular analytical technology can be applied to better understand water contamination and risks and add efficiency to the water quality management process and operational decision making. And it provides operators with improved information and tools to better evaluate and manage human health and environmental risks. The AWQC is the first water utility laboratory in Australia to apply NGS technology specifically to optimize water quality management. We have curated a custom-made DNA database of waterborne pathogens and have developed molecular testing methods that provide unrivaled pathogen testing results for a broad range of applications. Our NGS analysis has been recognized in the industry by winning the 2018 Water Industry Alliance Smart Water Award, and it has also been featured in the media, both on TV and print media on many occasions. And we don't just provide the testing service itself, but our experienced scientists work with our customers to provide advice and guidance on any investigation, project, or issue that needs to be addressed, utilizing our molecular services, ensuring the best outcome for the customer. Now, to provide you an overview of what we offer here, our molecular testing services include bacterial diversity profiling, or BDNA, and vertebrate diversity profiling, or VDNA, fecal source tracking, FST, E. coli phylogrouping, detection of cyanobacterial toxin genes, and whole genome sequencing. And I will be providing an overview of each of these. Of note is that all we do require in terms of sample volume is a 1 liter water or sludge sample collected in a DNA-free bottle, which we will provide, or a positive E. coli sample, for example, from a ColiAlert tray. We have produced a comprehensive fact sheet that clearly outlines the specific sampling requirements, and this is available upon request. Now I'm going to start off with the next generation sequencing, or NGS, our vDNA and our BDNA tests. These are NGS tests focusing on sequencing the DNA from vertebrates and bacteria. It's a very highly efficient method of DNA sequencing, and it's a high throughput DNA sequencing technology that is used to analyze water samples for all bacteria and vertebrates from the one sample and the one test. And we are using the Ion Torrent platform to conduct this. It detects all organisms by analysis of the DNA released into water, and is a screening tool for the identification of all of the organisms in that particular water sample. The DNA sequences that for each of the organisms that we detect in a sample are then compared against the reference sequences in our curated databases. And the results are presented in a user-friendly report in a PDF format, which includes a ranking of organisms detected in order of relative abundance. So the percentage of the total DNA of each organism detected. And I will show you an example of this in a minute. The report also includes dot point comments against the organisms detected, and we also provide the raw sequencing files on request. The bacterial diversity profiling, or the BDNA, is commonly used for the characterization of biofilms, nitrification events, filtration systems, and wastewater treatment plant processes, while the vertebrate diversity profiling, or the vDNA, is used for fecal source tracking, biodiversity surveys, and detailed catchment surveys underpinning these the catchment land use. It provides a better understanding of the sources, the types, and concentrations of biological contaminants. Catchment assessment, monitoring of discharges and exposure sites, all of these can be conducted using both our bacterial DNA and our vertebrate DNA diversity profiling. So how can this inform operational decision making? In a nutshell, this is a better, more targeted analysis enabling optimized decision making. The high efficiency and accuracy of NGS allows operators to implement rapid, targeted management actions for mitigation and control of any detected biological contaminants, thus significantly reducing the potential impact through early intervention. The detailed results also underpin targeted catchment surveys, implementation of catchment management actions, and information on the diversity and abundance of aquatic life in water sources, and detection and tracking of point source discharges. Although culture-dependent methods are extensively used, of course, for pathogen detection in water, these methods are limited by the lack of specificity. They do not provide a definitive diagnosis required for complex microorganism assemblages, and there's usually excessive time needed to obtain reliable results. And with the traditional culture methods we also need to know exactly what organisms we want to be testing for or looking for. So in a nutshell, our next generation sequencing service is efficient, it's accurate, it's run directly from water, sludge, soil, or wastewater with no culturing required, and all, of course, from a 1 liter sample of water. I'm going to show you a report. So this is where I'm going to focus on the BDNA report that we provide. So it's, as you can see, it's very visual with comments provided against the various orders detected in the sample. This detailed snapshot of the microbial community can have many applications, for example, to examine taste and odor issues in water, to detect the presence of surface water in groundwater wells, or to assess and track the health of microbial populations in water treatment plant processes. So in wastewater treatment plant processes, I should say. So what you can see here in the sample, you can see the diversity profile showing the top 10 bacterial orders. We report on all bacterial orders, but our report focuses primarily on the top 10 bacterial orders. So you can see there the percentage of these bacterial orders that were detected, and then also some explanatory comment in the box below. We also provide you with corona plots as well, in addition to this report, and of course the raw data. So the BDNA diversity, the bacterial diversity profiling has been applied in increasing the effectiveness of the wastewater treatment processes, for example, providing a better understanding and pinpointing and monitoring of treatment plant processes and issues, for example, sludge, bulking, foaming, and changes in the bacterial population composition ratios. We've also applied BDNA analysis in reducing nutrient levels in discharges, helping to prevent the occurrence of algal blooms, which can potentially suffocate and release toxins into receiving water systems. And currently, AWQC is helping customers using the NGS technology to gain a better insight into beneficial bacterial communities, with the aim of being able to grow these on a larger scale for incorporation into the treatment plant process to increase energy and chemical efficiency. We have also employed DNA in the past to locate the source of bacteria in bore water supplies, enabling operators to make informed decisions on the management of bores. Now, next, we are also offering rapid molecular techniques, fecal source tracking, and our cyano-detect. So if you want to determine fecal input, for example, if there is a sewage spill or inputs from septic tanks suspected into a creek or a similar scenario, then fecal source tracking is a rapid non-library dependent method that can discriminate human from non-human fecal contamination, and also bovine contamination. Our FST method utilizes host-specific fecal molecular probes and markers detected from the host-specific gut flora. And these markers can then be used as an alternative for fecal indicators, for example, like E. coli or fecal coliforms, because these markers make up a significant proportion of the fecal bacterial population and have little potential growth for growth in the environment outside of the specific host. And of course, they have this high degree of host specificity. So this is informative for risk assessment and for identifying problems in infrastructure or land use that contribute to degradation in water quality, septic inputs, as I mentioned before, sewage spills, and similar. Cyano-DTEC, this is a total cyanobacteria assay that detects and quantifies cyanobacteria in the sample and is reported as copies per microliter. And this only requires a very small volume of sample. We routinely apply this to 5 milliliters of sample. And the cyanobacteria toxin assay detects the genes responsible for the production of toxins. And we've covered microcystin, nodularin, cylindrospermopsin, and saxitoxin genes. And these are then reported as presence, absence of each toxin gene. Here you can see some examples for the fecal source tracking and the cyano-DTEC reporting that we provide. So in the example there for the FST, you can see that in this particular sample, a high level of general fecal matter was detected. However, no bovine and no human fecal input was detected. And in the cyano-DTEC example there, you can see that there were quite high numbers of genes detected for general cyanobacteria, so total cyanobacteria, and the cylindrospermopsin gene was detected at a gene copy count of 2,600 gene copies per milliliter. And then we've got the microcystin and the saxitoxin gene below the detection limit. Other molecular techniques that we do provide is testing from positive E. coli samples. For example, if customers submit a ColiAlert tray with a positive E. coli detect, then we can offer E. coli phylogrouping. Routine testing for E. coli, for example, via ColiAlert does not provide the information on the origin of the E. coli detected. So this information, of course, is particularly important when the identification of specific E. coli strains will determine the risk profile and water treatment options used by a water utility. And phylogrouping enables us to determine if the source of the contamination is of animal or environmental origin, which will then inform the risk assessment process and the water quality management actions that need to be implemented. For tracking of E. coli, and if more detailed information is required, then whole genome sequencing is recommended. And this provides, in addition to fingerprinting the individual isolates, this bioinformatics pipeline was developed to identify any genes associated with human health risk or the potential to form blooms. And E. coli strains responsible for elevated counts of blooms in freshwater reservoirs or recreational waters, they carry a capsule which may enhance the strain's ability to persist in the external environment. And typical environmental E. coli strains do not have pathogenic tendencies and therefore tend to be less of a health risk than the fecal-borne E. coli. And our E. coli capsule test that covers actually all known Australian encapsulated E. coli bloom strains, so we can offer that as well. Just to provide you a brief overview of the E. coli phylogrouping report and the risk rating. So E. coli strains can be separated into a phylogroup structure as shown in this matrix. And this presents, which at present includes the eight phylogroups you can see there. And I particularly would like to point out the B23 phylogroup, as this is predominantly carried by humans, and the risk of this type of E. coli to human health is high, as it may indicate, of course, a human fecal contamination event, and thus the potential for human infective pathogens to also be present in that sample. The other summary that I wanted to present you, this is my last slide here, is a matrix that summarizes all of the molecular tests and common applications that we provide here at the AWQC. So you can see at the top line there all the different molecular analyses that we provide, and then on the left-hand side some of the typical applications for these analyses. So from detection of E. coli blooms to biofilms and reservoir management and taste and odors and toxic algal bloom detection. So this matrix is available in our guide to molecular testing for waterborne pathogens, which is available via the AWQC website. This finishes my overview of the molecular services that we offer here at the AWQC for the improved management of water quality issues. Thank you.
Thank you, Torsten, for your presentation. So thank you. We've had some questions come through. So just a reminder to everyone, if you have got questions for the presenters as we go along, to pop them into the Q&A box at the bottom of your Zoom window. There's also an option if you'd like to pop it in the chat box. Our AWA support will also pop those questions in the Q&A box. But keep your questions coming. Today's webinar is obviously a fantastic opportunity to tap into the expertise that we have. So now we'll turn to Dr. Brad Clark from the University of Melbourne, who will be presenting some information on techniques for monitoring chemical pollutants in environmental samples using high-resolution mass spectrometry.
Great. Yeah, so I'm going to talk about some new analytical approaches for environmental monitoring. Just a little bit of background though to start with, and in many ways what we've been living through in the last 100 years is a chemical revolution, and we're going to continue living through that. And it's a bit of a shocking statement that I'm going to read to you, but every person in the world has synthetic chemicals in their bodies, and they're exposed to these chemicals from the moment of conception until death. And when we're not seeing any slowdown in the rapid introduction of synthetic chemicals into modern life. Now, the worst of these chemicals can accumulate in the human body. So they're referred to as persistent organic pollutants. So things like DDT, PCBs, flame retardants, and now PFAS. Now, the big issue with these chemicals, other than the fact that they're accumulating in our body, is that they can have a range of health impacts. So sublethal health impacts for those people that are exposed to them. And some of the worst health consequences can include impaired reproductive health issues, an impaired immune system, and neurological damage, which I think is one of the more frightening endpoints. So as on this screen, what you can see is this is from a study in Mexico where they looked at the exposure to 3-year-olds to pesticides, so side-by-side villages. And on the right-hand side, what you can see is that the children that were exposed to pesticides early in their life had a rapid decline in their cognitive functions, not able to draw a representation of a person. So I'm going to use PFAS as a case study. And so PFAS were a synthetic group of chemicals that are incorporated into a wide variety of consumer and industrial products. And that wasn't until 50 years later that that it was really documented that PFAS was present in almost all living organisms on the planet and distributed ubiquitously throughout the planet. And really, it's an advanced in analytical chemistry that allows us to have those discoveries. So what we have at this point is sort of targeted approaches for determining environmental pollutants, and it's a hypothesis-based approach as well. So what that means is that at some point someone's thought we better look for this chemical in the environment. Analytical techniques have been developed, and that's how we've determined that there's a problem. And there's a big gap in time here. There's 50 years as well. So so that part that analytical chemistry plays is quite big. Okay, so when we can also we're compounding this problem too. So if we have things like PFOS, PFOA, they were the prototype of this group of of chemicals. That carbon-fluorine bond is incredibly stable. It's very difficult to degrade, and these chemicals are persistent. They're environmentally mobile. They will bioaccumulate, and some of them are toxic, and they're being recognized as probable carcinogens. As a result, they get listed into various, they've been now listed as United Nations stock persistent organic pollutants. Now, this is also one of the realities we live in at the moment, is that the chemical manufacturing industry has no real mechanism to prevent or stop them producing thousands of different chemicals. So what happened was that we identified that PFOS, PFOA were issues, and that the industry then went and produced thousands more PFASs, many of which we don't know anything about what will happen to them in the environment, and also their impact. And so we have no ability to measure them, and we don't know what they do either. I'm going to use a case study to show you about how new approaches can help us understand environmental monitoring and impacts. So this is, some of you may be aware that...
Excuse me, Brad, sorry to interrupt. There appears to be an issue with your slides. They're not progressing. We're just on the slide that has the globe on it.
Right. Well, that's strange. Let's go back. Right, how are we going now? Can you see that?
That looks good.
And they're moving?
They're moving.
Cool. So here's the slides you missed out on earlier. So here's the PFOS, PFOA, the carbon bond, and here's where I was talking about the precursors. So this is the extended family of PFAS. So what we have is these per part of the molecule with fully fluorinated parts, and then we have this attachment here, which which will degrade in the environment to form the regulated compounds. And so we now have thousands of these types of chemicals being produced, or known of these known PFASs. Right, so this is what I was going to use a case study, and I'll rush through this, which is, it was a major pollution incident that occurred in Melbourne in 2018. We've just got a paper coming out on this now, so some of this information is present in that paper. Now, during this incident, there was it's a suspicious industrial fire where thousands of litres of industrial chemicals caught fire in the middle of the night, and it took 5 days for the Melbourne Fire Brigade to put out the fire. The runoff water ran into the local creek. And so at the time we thought, well, there's a reasonable chance that PFAS would be discharged from the MFB. And so we grabbed samples at the time during the fire. We grabbed them 3 months later, and we've also grabbed them 2 years later as well now. Okay, so site 3 is the point source of the fire on here. So, and site 1 and 2 are our control sites upstream, and site 4 and 5 are downstream, leading into Port Phillip Bay. And what you can sort of see that you start getting this bit of a discoloration occurring in the site 3. So there's this ash that was sort of getting washed into the samples as well. And so we did some what's called targeted analysis. So when you submit your samples for analysis, typically they're done using an approach called targeted analysis, and this is hypothesis-driven approaches to environmental monitoring, because you only can see what you're telling the instrument to measure. And so we have the capacity to measure about 60 different PFASs, which is one of the largest suite of PFASs in Australia. So we monitor a range of legacy and emerging PFASs. And the method we use is called isotope dilution. Now, isotope dilution is the gold standard for quantitation for these types of chemicals, and you use an instrument called a triple quad mass spec. Yeah, so there's just some of the details on our sampling approach. I'm happy to talk about that if anyone's interested. But okay, so here's the results of these studies. So what we found was that if you look at site 1 and 2, there's low levels of PFAS in that surface water. And then during the fire incident, there was a major release of PFAS. Now, it was very interesting now that we followed this up. The MFB says they don't use fluorine-based firefighting foams anymore. So that is not it's believed that that isn't actually the source of the PFAS. So the PFAS actually was probably stored in the chemical warehouse and was also then discharged during the fire. So we see really elevated levels, so 4,000 nanograms per litre. We also have a new cyclohexane PFAS in our suite, and it's the first time that that's been detected in the Southern Hemisphere, and actually at very elevated levels. So what you can see is you have this high spike of PFASs at site 3 and then declining as you go down further. When we followed it up, so it was 2 months later, not 3 months later, when we followed up 2 months later, what you see is also that the site is still releasing PFAS into the surrounding water body. And actual fact, when we followed this up 2 years later, we actually found still site 3, the point source, still had a statistically significant higher PFAS level than sites 1 and 2, indicating that even after 2 years, this site was still releasing PFAS. But the big question remains for environmental chemistry, environmental science is, like in particular in this case study, what actually was released into the environment. And one of the worst parts of this incident was that the firefighters who were exposed to all of this smoke now have many of them have now long-term health impacts. And so this is why we need a new style of analytical techniques for chemical measurement. Okay, so they're complementary, but you still need their complementary approaches. So you can't replace one with the other at the moment. So triple quad is typically the way that everyone does quantitation. It's only possible for predefined analytes. You won't see anything else. It's sensitive. And it has accurate, precise, and reproducible data. And so what we're going to talk about now is the time of flight. So time of flight is a really unique high resolution mass spectrometry approach where essentially you can see everything that's in that sample. Now, the big challenge now becomes we have massively large data files, and how do you interrogate that data file for actually determining what are the chemicals of interest? Okay, so this is our raw data for this site. So we actually determined there was about 50,000 unique chemicals that were found at site 3. What I'm showing you here is where we bumped up the filter. So we just said we want to just have a really high confidence threshold that these are actually unique identities. And so what we found was, we're finding that there's 8,200 chemicals that we identified from our study. And you can see that there's a significant amount at site 3. So what this is actually, this sort of analysis also is unbiased, and it just runs off the raw data. So this is a heat map, and what it's showing you is differences between the samples. And so you can see the red indicates that there's elevated or higher concentrations of particular unknown chemicals at this point in that sample. The other thing you can do with this type of work is look for differences, and this is called a principal components analysis. And so when you do this style of thing, this type of analysis, it's really useful for looking at what's been added to an environmental sample. Water is perfect for it, and a pollution event like this works really, is really tailored for this type of analysis. For example, you could do it. So you have flowing water, you have a wastewater discharge. You can actually determine through these types of analysis what chemicals have actually been added to the surface water from that discharge. And factory discharges, trade waste discharges, all of those things, it's possible. And so what you start seeing is that you get these clustering. So site 3 and 4 during the incident were very similar. Site 1s and 2 were very similar at both time points. And then we start seeing these sort of more spreads that site 3, 4, and 5 were still different at time point 2. The other thing you can do with this type of analysis is look just for unique identities. So this is a Venn diagram showing crossover of samples. So one of the things you want to try to do to figure out what are the main priority chemicals is by looking for unique features, and that's one of the things that we can do in this style of analysis. So to this point, what you're doing is only looking for differences and trends, so fingerprinting essentially. But what you can actually then do, well, there's two things to note. This file now becomes a historical record. So at any point now in this file, I can actually go and reinterrogate this and look for chemicals of interest. And I think this is going to be a really important game-changing aspect to environmental monitoring and regulation in the future. I think it'll be part of the compliance process that you must generate one of these files so that we can go and reinterrogate it and figure out when pollutants entered the system. So you do it by an approach called suspect screening, or this is one of the approach. And we have a custom database that we've created based upon the US EPA's PFAS priority list. You can screen it against all these other chemicals too, pesticides and typical water contaminants. And what you're actually doing is looking for matches between the spectra and your database. Okay, so this is that same data file. We actually compared whether we found the things using this new approach with our targeted approach, and indeed we did. And what you can also see is all of the things not in yellow are all of the new emerging contaminants that I don't think many of them have ever been reported in Australia before previously. Okay, so really at the moment what we need is it's a cycle. You've got to do your targeted work. You need to know about the regulated chemicals, but potentially they're not the most important or relevant in terms of pollution events. And that's why you'll need to do some non-targeted analysis and suspect screening. The challenges we face are significant. There's 163 million unique chemicals registered in the database, CAS database, and we have a serious challenge in the water sector on our hands about figuring out how to best manage this issue. Thank you very much.
Thanks, Brad, for your very interesting presentation. And just a reminder to everyone to keep the questions coming in the Q&A box, and we'll come back to those at the end of the session. Okay, so now I'd like to hand over to our next presenter, Dr. Barbara Drigo, who will be discussing examples showing how molecular tools can be used to provide insights to support risk assessment and health management.
Good afternoon, everybody. I'm Barbara, and I'm working at the University of South Australia, as previously mentioned. And today I will talk about using molecular tools to manage known and emerging risk in wastewater and water products. Monitoring and managing waterborne disease is a critical aspect of biosecurity. It plays an important role in minimizing the risk of infectious disease entering, emerging, establishing, and spreading in Australia. This helps protect the Australian population, food security, and economy. Our biosecurity system is under growing pressure. From the number of people and goods coming into Australia. And this number are forecast to further increase, as shown here in the slide for 2025. Globalization, complexity of international supply chain, and changes to the Australian operating environment, including those caused by the current pandemic, COVID-19, are making biosecurity risk management more challenging. Nowadays, a range of techniques range of techniques is used to help manage biosecurity risk. And during this presentation, I will focus on genetic tools used for water monitoring. One important example is wastewater-based disease surveillance. The World Health Organization uses wastewater surveillance to uncover polio outbreaks and to assess immunization coverage in resource-limited countries. Because of the urgent need to monitor community-wide spread of a coronavirus pandemic, wastewater monitoring has been extensively deployed to monitor the spread of infection in cities, neighborhoods, and important locations such as aged care home, schools, and correctional facilities. Wastewater surveillance can reveal an emerging outbreak in an area before it becomes evident through clinical testing, as you can see in this slide. And this means that it can be used to understand the human geography of infected and unvaccinated community, which is really important nowadays, and can facilitate intervention such as increasing the level of testing, limited area lockdown, tailor public health messaging, and suggest convenient access via pop-up vaccination site. Beyond vaccination support, this approach can and will become a part of the fabric of public health for cost-effective, near real-time surveillance of general population health. So, wastewater contains a highly complex mixture of microorganisms. Here in this slide is an example of a bacterial network. So each dot represents a different genus of bacteria, and you can see that the influent in red and the effluent in blue have characteristically different microbial community, whilst the sludge, anaerobically digested, primary sludge, activated sludge, they share a common community. Luckily, we can use molecular tools to cut through this immense microbial diversity and focus in very precisely to target specific genetic marker, such as genes linked to specific disease or other health and biosecurity risk we wish to monitor. And in here you see the example of coronavirus. So to develop disease or health biomarker, we first need to characterize the disease using various molecular tools. Here in this slide, there is an example of how biomarker for coronavirus monitoring were built up. So the first step was to identify and visualize the disease agent. And here you see a picture of the coronavirus. Understand his phenotypic characteristic, and in this case, the corona outside the spikes, and then sequence and determine how many variants are present. And so you see a phylogenetic tree which couple all together the Oceania, European, American, Asia, and Chinese and African strain. Once we have this genetic makeup, so we know the genetic characteristic of the agent, the disease is possible to build up primary techniques that target conserved genetic regions so that it can be detected using molecular assay. We currently use next generation sequencing, as Torsten mentioned, real-time PCR and real-time LAMP to detect the quantity or presence and absence of pathogen, opportunistic pathogen, and antimicrobial resistant genes. A number of other technology have either been developed or are in development. For example, in our lab, we use digital droplet PCR to detect infectious agents, including coronavirus, in high throughput with high sensitivity. The benefit of the digital PCR is its accuracy in detecting very low gene number, and the digital PCR does not need standard. Other techniques such as cluster regularly interspace short palindromic repeat and SARS-CoV-2 proteome peptide microarray are also available and will be in the future applied to disease agents. Wastewater-based epidemiology can be used in responding to another emerging public health catastrophe, this being antimicrobial resistance, or IMR. Antimicrobial resistance occurs when microorganisms change and adapt to resist stress, and its impact is the medication used to cure infection. For instance, the antibiotics become ineffective. So what we do now? So without action, it is estimated that by 2050, 10 million lives a year and a cumulative $100 trillion of economic output are at risk due to the rise of drug resistance infection. Therefore, it's imperative to globally enhance antimicrobial stewardship in order to maintain the efficacy of available antibiotics while we are seeking for alternative therapy. This is logically more urgent in the case of antibiotics that have been considered last resource, which is a label that we give to the antibiotics when they are of vital importance in combating critical infection. So one example of a critical antimicrobial resistance is colistin resistance. As you can see in this slide, the use of colistin, also as polymyxin E in human medicine, has historically been restricted because of its toxicity. However, in recent years, colistin has become increasingly important as the last line of defense against fatal infection caused by multi-drug resistance organisms. So the identification of the first mobile colistin resistant, or MCR gene, was the variant 1, MCR1, and in 2015 triggered a rush of MCR screening reports. Subsequently, 9 other MCR family genes and their variant have been described. And in these slides, we can see that the first variant of colistin, so MCR1, has spread mainly via people traveling in most of the world, and the other variants are slowly colonizing the world due to people movement. So to monitor this issue, so the antimicrobial resistance and the presence of and dissemination of antimicrobial resistance, in our lab, we use next generation sequencing, qPCR, and digital PCR. In these slides, you can see one of our recent articles, and here we quantify the presence of antimicrobial resistant genes in reclaimed water or wastewater treatment plant effluents to evaluate the effect of treatment and disinfection. We also evaluate the capacity of the organism to carry antimicrobial resistant gene to regrow in the environment post-disinfection. This has shown that some organisms carrying antimicrobial resistant gene can regrow post-disinfection and may have the capacity to proliferate. In particular, in these slides, you can see the RNA, so the metabolically active microbial community, after 7 days of incubation, are growing a specific antimicrobial resistance, and they are resuscitating, is called this process. So they are basically able to regrow in the environment, put in optimal condition. Similarly, to evaluate potable water quality and the effectiveness of disinfection technique, we can quantify and characterize human opportunistic pathogen using qPCR again and next generation sequencing, whole genome sequencing, and traditional microbiology techniques such as plating on selective media. So in this study, our lab monitor how the quantity of Legionella, an important respiratory human pathogen, was affected by the installation of a disinfection booster system in the potable water distribution system of an hospital and an aged care facility. And what you can see is prior the installation of this system, the level of Legionella was quite high, then slowly started to decreasing, highlighting that the disinfection system was working properly in the potable water distribution system. In the same facility, we also monitor the potable water quality using amplicon and shotgun sequencing, and we analyze the total microbial structure in the pipe biofilm. This analytical approach was used to understand what other human opportunistic pathogen and antimicrobial resistant genes are present in water network room by room, and how we prevent infectious disease. And so in here, you can see all the different colors represent different phyla, so a family of organisms, and how they change after the introduction of the booster system, which was cleaning the water. To conclude, effective water monitoring is essential to underpin Australia's livelihood. A safe water supply is critical to protect human health, the environment, the economy, tourism, trade, and agriculture. So it is crucial that we keep on developing systems that support early detection and early warning of biosecurity and infectious disease risk. This will allow us to respond much rapidly and efficiently to future outbreaks like we did for coronavirus and avoid economic losses. So to conclude, I would like to acknowledge our laboratory and Erika and Gianluca and Sotos and Sam and Michael, and also the Colossus Initiative, Water Research Australia, SA Water, Melbourne Water, and the Government of South Australia. Thank you.
Thanks so much, Barbara. A very interesting presentation on how molecular techniques can be used to help us understand both the actual and emerging public health risks associated with pathogens and antimicrobial resistance. We've had some great questions coming in, so continue to post your questions in the Q&A box. If I could ask if you could actually just indicate the speaker for the questions so we can keep track of that, that would be great. Okay, so last but not least, we have Karen Rouse, the Chief Executive of Water Research Australia, who will now present some fascinating information drawing on case studies, including Water RA's collaboration on the sewage surveillance of SARS-CoV-2 that Barbara referred to, the Colossus program. And Karen will illustrate the importance of dialogue between developers of tests and end users of tests during the test method development process, and importantly, approaches to gaining regulator acceptance of new tests and technologies.
Thanks, Karen, and hello again, everybody. As Karen said, my talk is more around engagement and how to ensure that the new tests and wonderful capacity and capability that you've heard about is able to be used to deliver value in the Australian water industry and beyond. So part of the importance of achieving this relates to who's involved in the process. And here at Water RA, what gives us the ability, I suppose, to talk on this topic is the profile of our membership, we have members from water utilities, from government departments, including health regulators, all of those around the country, as well as EPAs, research providers, laboratories, and consultants. So what we find is it's very effective when you have all the people in the room or across a problem that are needed to both understand the problem, develop the solution, and then implement what you find. So I would encourage you to keep track of all those different parties when you're thinking about new test methods or new technologies. So one of the things that we've done in recent years is to look at the value of research. And I use the term research, it could be for test method development, for example, within the water industry, and to talk with those participants in the water industry about how to ensure that you do get the most value and the most benefit to customers, members, community from the work that you're doing. And we developed what was called the Good Practice Guide to Delivering Value from Research. And as part of the study, the research that was undertaken to develop this guide, we consulted widely across the industry to determine how to maximise the value and the impact that is achieved. And what you'll see from this diagram here is that you can divide the research process into 4 stages, and it's quite often shown as a linear process, but we think there's value particularly in representing it in the way that you see here, because you can start with the plan and propose stage, which is where you have the ideation, you explore, well, what is the problem, what is the opportunity? You can then move into the develop and organise, where you work out how you might go about scoping the research. And then you actually undertake the research.
Excuse me, Karen, your slides are not progressing, so I wonder if you could just check.
Okay, thank you. I'll just resume share. Try this. Can you see the good practice life cycle now?
All good, we can see that.
Okay, let's hope it continues. And so what I wanted to draw your attention to was this teal colour here, which is the phase where we engage and achieve impact. And you'll notice that it does actually envelope the whole process. And this is to illustrate how important it is to engage with the end users and the people that are going to be able to ensure the impact occurs right the way through the process. And in particular, in relation to the topic we're considering today, I'm thinking of regulators, because if they do not recognise or cannot see the way to use the test methods in terms of achieving their health or environmental outcomes, it'll be very difficult to see the uptake. So that early engagement, whereby you can discuss what the new capability might be or what you're setting out to achieve, and then there can be a dialogue around, well, how could it be used, how could it be integrated? And if there is a need for regulatory change, you've actually got a head start and time to be thinking about that before you get round the cycle to engage impact. So I realise that it may be difficult to get the regulators' attention at times. They're very busy people, particularly health regulators at the moment. But it is possible to sort of put yourself in their shoes and think about what their view might be. And one way to do that is to consider the regulatory frameworks within which they operate and we all operate within the water industry. So just checking that you've got my next slide. So please tell me if you haven't. I've now moved on to regulatory guidance.
All good, Karen.
Thank you. So here, for the majority of water quality, both from a health perspective and an environmental perspective, it's guided by the National Water Quality Management Strategy. And beneath that sits a whole series of guidelines relating to drinking water, recycled water, and water discharges to fresh and marine environments. So this guidance is used by all states. The actual frameworks that are in place. The legal frameworks vary somewhat between states, but they're all guided by these particular documents. And then the additional one there is the National Environment Protection Measures, which, unlike the guidelines above, are actually mandatory for the states to follow. So one of the examples that I wanted to take you through was to look at the water vow protocols and framework that was developed by the Australian Water Recycling Centre of Excellence, and has since, the IP has passed to Water Research Australia. And this framework was designed as a nationally consistent approach, the validation of water treatment technologies, to enhance confidence in managing water treatment and drinking water supply. So this is very important, and it's really based around the multi-barrier approach and understanding what kind of barrier to pathogens and potentially in future chemicals a particular technology can deliver. There are some protocols already developed around chlorination, membrane bioreactors, reverse osmosis and nanofiltration. And they're developed following a series of elements that are worked through for each technology. So whilst we've looked mainly at test methods today, I did want to highlight the fact that there are frameworks out there which have been developed and are very strongly supported by our health regulators. So if you are looking at new technologies and you know that down the track you're going to need approval from the health regulators for a new recycled water system, for example, then the water vow protocols are one tool that can be used to potentially ensure and accelerate that approval. So it's still a developing suite. So a number of those protocols were developed by the Water Recycling Centre of Excellence. And we already know that there is interest in a further few protocols, particularly around for pathogens in microfiltration, ultrafiltration, combines ozone and BAC, and granular media filtration. But another exciting potential use of these protocols is to develop them for chemical contaminants of concern. We've heard from Brad how the numbers of chemicals of concern are rapidly multiplying, and as is our capability to detect them. So at the end of the day, we want to be able to be sure that our treatment processes are able to remove those that are of concern and potential health damage for particularly humans, or it can be for livestock, etc., with recycled water schemes. So we are looking to develop protocols that might be applicable for chemical contaminants of concern. And then I wanted to talk about acceptance of new test methods. And here I'd like to sort of speak a little bit about the Colossus program that has been run by Water RA, which is collaboration on sewage surveillance of SARS-CoV-2, and has involved all of the health departments from around Australia, many of the water utilities, many of the laboratories and universities, who have all come together to, in a very rapid time frame, we've termed it real-time research, to develop methods for detecting fragments of viral RNA in sewage from the SARS-CoV-2 virus. So in various laboratories, there are different test methods being developed. They've all got some similarities, but they are also have some variants. And so what the health departments are asking is that we can be assured that tests in one state are, the results are as valid as results in another state, etc. So as part of the Colossus program, we've developed an alliance with Vidral and the National Measurement Institute to arrange for testing to occur for the different processes used at the different laboratories. We've got 13 laboratories participating in this testing. So I suppose it's a standardization validation test. And the reason there's so many is one of the things we've learned from the pandemic is the need for local capability. There were issues early on in terms of transport of samples, transport of reagents, availability of reagents. So it is a situation where it's not competition between laboratories, it's about collaboration and building local capability and capacity. So as part of this process, gamma irradiated SARS-CoV-2 material was produced by Vigil in Victoria and then concentrated and quantified by NMI so it could be used as a spike in wastewater samples by the participating labs. The materials have been distributed to those labs with a study protocol so that everyone will use the same number of replicates, for example. And then the results will be provided to NMI for collation and production of de-identified Z-scores and interpretation of the sources of measurement uncertainty. And this report is due around the end of April, and it will help the laboratories themselves to see how they're performing in comparison to other laboratories, and also to see what the relative differences are around the country. So it's a good example of collaboration, involvement of the health regulators at an early stage. They have been partners in Colossus from day one, and it's really illustrating how we can progress very quickly when all the parties needed to deliver something are involved from the outset. So my last couple of slides, really about what's next. This graphic is a timeline that describes the water quality incidents, issues and events that have shaped our science to date. It starts with the origin of the CRC water quality and treatment back in 1995 and comes through to the present day. And whilst there's a lot of information in it, you will notice that the dark blue circles, which represent regulation, are all only really occur in the first half, which is a sign that all of these guidance documents that I mentioned at the start are all either undergoing review or due for review. So it's very timely now for that dialogue with regulators around the new capability and capacity and how that can be picked up and to the benefit of the guidance that is updated in the coming months and years. So I just will also point out that this graphic was from our publication Health Stream, which is a quarterly publication, which provides water quality insights for today's professionals. And it really is the place where those multi-end user and researcher perspectives come together to describe how impact can be achieved and what has happened in relation to water quality incidents and what's happening, not just in Australia, but globally. So anyone whose organisation is a member of Water RA has access to this as part of their membership package, but anyone else is welcome to subscribe. So that was all I wanted to say today. Thank you, and look forward to an interesting panel discussion.
Thanks, Karen. Thanks very much. Well, we've heard from some great subject matter experts today who've presented information about how the latest testing technologies and methodologies really assist water quality managers to optimise water and wastewater treatment processes, and just also the importance of connecting in with our regulators as well as we're developing those new techniques, all about protecting public health and the environment. So a big thank you to our panelists today. So thank you also to those of you that have been posting your questions. So we're going to now move into the moderated Q&A session. So I'm just bringing the questions up here. So we've got a couple of questions that have come from Steve Delides, and I think these are directed to Torsten. So Torsten, a question around can the molecular techniques differentiate between DNA from live versus dead cells?
Yes, thanks. That's actually a very interesting question. We do need live cells actually. We can pick up DNA from cells that are dead if relatively recent. So that's the proviso there. So it needs to be relatively recent, but best of course is fresh material or material that is if they are known to be dead cells, that it's very, very recent. So that's in a nutshell, that's what we need to analyze successfully and get valid results.
Okay, and Steve also asked a question about whether there's any specific holding time requirements to be able to support an optimal valid test result.
Yes, look, we're looking at about 48 hours from sampling to receipt here in the lab. However, what we suggest at various times is that samples can be filtered in the field and then frozen on specific membranes, and we can provide guidance on all of that. So that's not a problem, but they can be filtered on site or somewhere in the workshops. Say, for example, it needs to be under specific, wearing specific DNA-free gloves and all of this. So, and then they can be frozen and then sent to us on ice, so that if it is expected to be a longer transport time.
Okay, and a question for Brad from Phil Weatherall. How practical is the time of flight mass spectrometry to implement at a small utility-based laboratory?
In some ways, Phil, that will probably depend on how rich the small utility is. I'll assume that the small utility is probably not rich, so not particularly practical to have an in-house time of flight, but there are commercial labs that do offer it. Eurofins, for example, does offer it. I suspect in the future that it will become a regulatory requirement for effluent discharges and for biosolids that are being applied to land. And there'll be a requirement on keeping that digital record so that we can go back in time and see if there's any incidents, which do occasionally occur from those practices, and be able to identify the chemicals that were problematic.
Thanks, Brad. And a couple of questions for you, Barbara. One from Steve again was asking whether the biofilm profiling via a direct swab was via a direct swab or downstream water sample.
It was by a direct swab, but also we collect the downstream water. So we do both analyses.
Okay, and another question from Shannon McBride has asked whether the project report for ECAS4 is available.
I will let Erica reply to that. Is she online?
Yes, I think it can be. If you want to contact me, I can put you in touch with the people who own the report, and we can make sure. But I'm sure they'll be happy to share it.
Okay, thanks, Barbara and Erica. Got another question for Torsten. Torsten, if you can expand upon the use of NGS bacterial diversity profiling on reservoir management.
Yes, so the bDNA, it's very useful to look at temporal changes in microbial communities in reservoirs. For example, if you know there's some upstream catchment land uses that provide or that might affect water quality. So you know, if you do a baseline and then regularly do a whole BDNA screen, and that because that will provide you and characterize the entire microbial community, and you can then see if there's certain bacteria of concern that increase or decrease depending on what the catchment land use, if there's any changes up in the catchment. So so it's good for trending microbial communities, also for recreational use. So you know, if you have recreational activities happening, and if there's any changes in the microbial communities associated with that, for example. And you can also link that with the fecal source tracking as well, of course, as well. So that will all contribute to overall catchment and reservoir risk management. And so it's certainly a very, very useful tool, but I would almost recommend that it's also been that it be run in conjunction with vertebrate DNA as well, to provide you, at least initially, to provide you an input, an overview of what other inputs are coming into a specific reservoir from further upstream. You know, if there's any dairy farming or who knows what happening in the catchment.
Okay, thanks, Torsten. We've got a question, Barbara, I think this one's for you, from Bowen. Do we have any indication on how much COVID-19 virus is going into our wastewater treatment plant and how much is still in the effluent stream?
Oh, yeah, this is a very good question. So there has been done study, and there is one, the first study was from Thomson in water research in 2020, which they evaluate the amount of population shedding, the amount of infection in the wastewater treatment plants, and they calculate how much coronavirus was possible to recover in the influent at the start of the wastewater treatment plants. And it was the amount of coronavirus detected was very similar to, and higher actually, than the amount detected from the health system, from the public health. And this is because mainly the influent can detect many people who are not yet tested. About the effluent, it depends in which country you live. So if you live in a country which is industrialized, who has a proper wastewater treatment plant with disinfection treatment, then the coronavirus is highly unlikely to survive through the wastewater treatment plant. Is actually deactivated almost immediately as soon as entering the wastewater treatment plants. If you live in a country which has doesn't have treatment for raw sewage, for instance, some third world country, or they have limited treatment for raw sewage, they find out that actually coronavirus survives and entering the environment. So it really depends on which country you live.
Thanks, Barbara. We've got another question's come in for Brad from Shannon, and it's just regarding the time of flight. So Brad, Shannon's asked that you've indicated that regulators may require this test for sewage treatment plant effluents as an example, but the results look a bit complicated. So who would likely interpret these reports, or how would the reports be provided by a laboratory to assist interpretation?
Sure. Just like most of these techniques, they'll need to be run by technical experts. So, but like if you think, like with the example I provided, it was 50 years between the release of the chemical of PFOS and until it was determined to be problematic. Now, with the time of flight data, we could actually, if we had those 50 years of records, we could actually go through and figure out exactly when it was released into the environment. It's actually quite simple in a way. Once you actually have the data, it's actually quite simple to like interrogate it for a compound of interest. All we need is a formula, and then we can go and look for it. The data files are large, so it's not really feasible So there are two ways that you can go about it. I can probably give you your top 10, and also we can do a suspect screening approach. So as I say, I do think at some point this will become a toolbox that will be used for environmental monitoring and protection. And also I imagine at some point there'll be a digital archive that will at least be available to people in the sector, where we can go back and look at, even if it's just annual samples, about what was present. And if you think about some of the events that can happen, like there's recycled water occasionally will kill crops, for example, and it's difficult to determine what that actually was. With the time of flight, it becomes, once you have that data file, it's possible to eventually figure that out. And also when you combine that with the temporal nature of the collection that's possible, it's going to transform how we track down problematic chemicals.
Thanks, Brad. We've got 3 minutes left. I've had one more question come in actually for me around whether the contact details for our wonderful speakers can be provided. As provided the speakers are okay with that, we can arrange that. Okay, thanks, Paul. I've had a question come in from Paul Sherman. The answer about the survival of SARS-CoV-2 virus differs from other expert advice. Due to the nature of the envelope, the virus does not survive very long in the environment. We are only detecting RNA fragments in sewage. So please comment on that. So that's probably for you, Barbara, that question.
Yeah, that's correct. So in Australia and in all industrialized country, because our efficient way of cleaning, the sewage is actually destroying completely. And also at the enter of the influent level, you can find a RNA fragment of coronavirus because it's an RNA. So, and RNases are everywhere. So as soon as you shed, you basically, the capsule of the virus can be destroyed. And as soon as that enter your stomach, actually, it can be destroyed. So I would say, yes, RNA fragment in sewage. We also find only RNA fragment in sewage. However, there are several studies done in a third world country, which actually coronavirus is surviving and entering the river systems. And they are published. I can send you some of the reference of those.
Thanks, Barbara. 1 minute left. So if there are any last minute questions, if you'd like to quickly post those. Otherwise, just thank you, everyone. Thank you to the panelists today, our amazing panelists, and thank you to everyone that joined the webinar. As we said, we've recorded today's webinar, so that will be available, and we'll also make the contact details of our amazing panelists available as well. So thank you everyone.